Hello, welcome to the the Headwaters Science Center 3:30 show. I'm Spencer Potter, and today we're going to be exploring the world of cells. What kind of cells are there, and really what do they all have in similarities? What kind of similarities do they have? So, in broad terms, we have two different types of cells. We have our prokaryotics, which have no membrane-bound organelles, and an example of those would be like bacteria, single-celled organisms. And then on the other side of the other one would be eukaryotic cells. They do have membrane-bound organelles. An example of eukaryotic cells would be you and me. You and I, like a fun way to remember prokaryotics and eukaryotics is like pro and no rhyme. So prokaryotic cells have no membrane-bound organelle. You and I have eukaryotic cells. So what are organelles? Let's take a look at our cell example right here. You can see that there is a lot of stuff going on here. It's not just one big blue ball. You can see a nucleus, you can see mitochondria, you can see different types of endoplasmic reticulum, ribosomes, maybe there's a Golgi apparatus in there, lysosomes. So those are all examples of organelles. Now, from what we just talked about, we said that eukaryotic cells have membrane-bound organelles, and you can tell that there's definitely a lot of organelles situated in specific spots in this cell. So we could make a pretty good assumption that this is a eukaryotic cell. One thing that isn't really labeled on this cell is what encases the whole cell. What is holding together all cells? Because even though they're different categories of cells, they do have some similarities. Some similarities are they all have cell membranes. So they all have cell membranes. They all have DNA, and they don't have DNA in the same form, but they both have DNA, and they both perform life functions. Okay? So if an organism is going to be alive, they need to be able to respond to outside stimulus. They need to be able to reproduce and use energy, metabolize. So they're all able to do that. So let's take a look at similarities that they do have. The cell membrane. Cell membrane is referred to as a phospholipid bilayer, and a phospholipid bilayer is pretty self-explanatory. Phospholipid bilayer. So if you take that word apart, you got phospholipids. So you got a phosphorus polar head here, and then you got two tail lipids that are non-polar. So if you line a bunch of them up, you get a phospholipid bilayer, because the two ends of the non-polar lipids, tails, they line up and form a nice membrane. This gives the cell flexibility, gives it, allows it to absorb things too. Permeability. I was going to say permeability, but it seemed like too close to flexibility. But permeability, flexibility, so it contains the cell. That is the barrier between the outside environment and the inside environment. And in the inside environment, all living things are all trying to reach a homeostasis. And without a phospholipid bilayer, it would be pretty hard to establish a homeostasis with the outside environment. If we weren't being able to control what comes in and what comes out.
What's homeostasis?
Homeostasis is forming a balance within an environment. Like in our cell, we need a certain amount of salinity or salt. So our cells work to either bring in more like nutrients or salt, or if we have too much salt, it'll work to expel them into the outside environment. So it's all about balance. Homeostasis is all about balance. And we'd mentioned that the cell membrane is moving things in and out. But it's not really. The cell membrane isn't always moving things in and out. Sometimes things just can move without using any energy. And that's where we come into what's called a passive transportation. Passive transportation is this great thing that requires no energy. So we have three different categories of passive transportation. We have diffusion, facilitated diffusion, and osmosis. So I said diffusion twice, and I did not make a mistake. I did say diffusion and facilitative diffusion. And for demonstration, I have a super awesome drawing here that you can tell that I don't have a very good future in art, but what I've tried to represent here is the phospholipid bilayer and some different particles here. And they're different sizes. So diffusion is just moving across the cell membrane without any specific spot. So that's where you get like your oxygen, your carbon dioxides. Those are just straight up diffusion. Diffusion is they're just passing right through the membrane from high concentration to low concentration. Diffusion doesn't just happen in cells. Diffusion happens in the air we breathe. Have you ever been in another room and smelt someone cooking bacon in the kitchen? That would be a great example of diffusion. The particles are leaving the concentrated area of the kitchen, flowing down the hall, under your door, into your bed, and into your nose. So that would be an example of diffusion. So they're just going right through. They're able to pass right through the cell membrane. And that typically happens with not like very small molecules, not and like they're not equal. They're equal. You know, they're equally balanced molecules. That's what I'm trying to say. They're equally balanced molecules. Not both. Well, yeah. So then we have facilitative diffusion. Facilitative diffusion, if we take that word apart, it has facilitative. So we need something to facilitate that type of diffusion. So in our drawing, we have these embedded proteins. Line right here. Boom. Embedded proteins. And sometimes those proteins are just there to move molecules from the inside to the outside. And again, we're still talking about passive transportation, where we're not requiring any energy. So facilitated diffusion is just saying that we need this molecule maybe is polar, or maybe it's just too big to pass through the cell membrane by diffusion. And then osmosis. We have osmosis. And that's dealing with water. Water is moving from high concentration to low concentration. And we have a super awesome protein called aquaporins. Aquaporins. If we take apart that word, we got aqua and then porins. Aquaporins, so aqua refers to water, and porins refers to holes, pores refers to holes. So basically the word says water hole. So aquaporins is an essential protein in moving water from high concentrations to low concentrations through the process of osmosis. And that all has to do with moving molecules from high concentration to low concentration. And but that's not always how things work. Sometimes we need to move things the opposite way. An example would be, imagine that you're kayaking down a river. When you're going with the current, that would be passive transportation, because you're just going along with the flow of the river. You don't need to do much work. But then all of a sudden you get to your destination and you got to turn around. Now you're trying to paddle up the river, and that's where you have to start expending energy to work against that current, to move up further back up into the stream. And that's where we get into active transportation. Active transportation is just like that kayaker that needs to paddle back up the river. It requires energy, and oftentimes it's in the form of ATP. Then it's adenosine triphosphate, I think is the full word of ATP. And again, just like passive transportation, we're going to focus on 3 different types of active transportation. So the ones that we have on the board are endocytosis, exocytosis, and karyoproteins. So just from the first 2 words, endocytosis and exocytosis. So if we just had to take a guess in which direction these are transporting molecules, endo and exo. So I'm going to say exocytosis is just like exiting the building. You're exiting the building. Exocytosis is expelling waste and material outside of the cell, past the cell membrane.
Or like an exoskeleton.
Yeah, like insects have exoskeletons. And endocytosis has to do with bringing nutrients and molecules into the cell. And karyoproteins are just like the aquaporins, except for you need a little bit of phosphate, or a phosphate group, to break off to give that protein enough energy to open up, squirt a little bit of some molecules out, vice versa, in or out. So endocytosis is actually just a modified vesicle. Vesicle is a organelle, and a vesicle is basically just a bubble of phospholipids, and they form a group around whatever substance that they are trying to bring in, and they bring it in and distribute it into the proper areas of the cell. And endocytosis, so that is endocytosis. Endocytosis is bringing in molecules through some sort of vesicle, like absorbing the whatever the molecules that they need. Reverse that, exocytosis is the exact same thing, except for in the opposite direction. Endocytosis is a vesicle full of waste or water or anything that the cell feels like it doesn't need. It's going to be thrown into a vesicle and transported out of the cell. And once it gets to the cell membrane, it will open up and release the particles into the outside environment. And Then we just have carrier proteins. Carrier proteins work and look similar to this example. They're protein embedded within the cell membrane, and they work to open and distribute molecules in the proper direction, which typically is against the flow concentration. So active transportation is taking a molecule that's in a low concentration and forcing it into a high concentration. Passive transportation is taking a molecule from high concentration and allowing it to go to a low concentration. What else did I say?
Well, and we've talked about some of this stuff in one of the other shows we did. So if you're interested in seeing this kind of in action, one experiment that you can do at home is you can take a glass of water and pour some salt in it, and then put a stick of celery in the salt and let it sit overnight. And what you'll see is that eventually that celery is going to get kind of weird and wilty. And the reason for that is because of osmosis. The water leaves the cells from an area of basically low salt concentration to an area of higher salt concentration, tries to dilute the water with the cells, or tries to dilute the salt further with the water in its own cells, and makes all the cells collapse and it gets all limp and wilty. So it's an experiment that we do here on occasion when we're talking about specifically water.
Yeah, and like Ryan was saying, that's a great example of a hypertonic solution. When we're referring to osmosis, that's you have to take into account hypertonic solutions, hypotonic solution, and isotonic solution. So hypertonic solutions, and a great example, just like Ryan said, would be putting a cell in a salt solution, and that would be a hypertonic solution because there'd be a higher concentration outside of the cell than inside of the cell, which would force the cell to be shrivel up. A hypotonic solution, if you put a cell in a hypotonic solution, which means that the concentration is higher in the cell than outside of the cell, you'll see a swelling of the cell. And in an isotonic solution, it is matched. So if you ever were in a hospital or ever watched any types of hospital shows or ER shows, they're hooking up people to IVs, and the IV bag may look clear, may look just like water, but it's actually not. It's actually a solution of saline, a saline solution. Maybe you've heard of that, and it matches similar to the same salinity as our blood and acidity to our blood. That way, when you're injecting or trying to replenish fluids within your body through an IV, you're not just lysing all your blood cells and just making them too big if you just pump straight water that we drink into your blood cells. And just like everything needs to maintain a balance, which we used that word homeostasis in the beginning of the episode. So all these play into the factors of maintaining a homeostatic environment within the cell. And you may think, wow, that is pretty crazy. Where does that even happen? Well, just look around. Anything that is moving, anything that is reproducing, you and I, every cell in our body is going through a process of passive transportation and active transportation. So, if you would like to come explore cells with us, we have many microscopes, many samples to examine, microorganisms and different types of plant cells, animal cells, prokaryotic cells. All of those are available here at the Headwaters Science Center. And you can come in Monday through Saturday, 9:30 to 5, and then stop by after lunch on Sunday 1 to 5. Well, I'll see you guys next Sunday. Have a good week, and I'll see you at the Headwaters Science Center.